Hello friends, it is well known that humans and great apes differ in chromosome numbers. Humans have a total of 46, which is a pair of 23 chromosomes, while great apes, particularly chimpanzees, have a total of 48, which is a pair of 24 chromosomes. Humans therefore have one pair of chromosomes less compared to chimpanzees. If the so-called evolutionary consensus on human primate evolution is correct, which assumes common descent, it will need to explain the disappearance of one pair of chromosomes in humans relative to chimpanzees. In other words, there cannot be an unexplained discordance in chromosome numbers if common descent is to be assumed. So, where did the chromosome disappear in the history of human primate evolution? Well, the difference in chromosome number is claimed to be due to a supposed prehistoric end-to-end fusion of two acrocentric chromosomes corresponding to 2A and 2B in chimpanzees. This putative fusion occurred in a human chimpanzee ancestor to form human chromosome 2, which led to 46 as opposed to 48 chromosomes in humans. This is considered to be a key piece of evidence to support common descent between humans and chimpanzees, and is referred to as the chromosome 2 fusion model, or in short, the fusion theory. The idea of fusion first emerged in 1991 when an alleged fusion-like region was discovered on human chromosome 2, although the original hypothesis can be traced back to 1982, which is almost a decade earlier. Impressed with this hypothesis and the preliminary reports that followed based on indirect lines of speculative evidence, evolutionists began to consider the chromosome 2 fusion model to be a very strong evidence for common ancestry between humans and chimpanzees. Nonetheless, it was the loveliest but not the likeliest explanation based on the available scientific knowledge. Eventually, as time went on, it led to the belief that the prediction of common ancestry has been fulfilled and that the case is now closed. But is that really true? Fusion theory has crashed into insurmountable problems ever since. For example, geneticist Dr. Jeffrey Tompkins has written extensively on this topic and makes some plausible scientific arguments against the fusion theory. Let us closely examine a non-exhaustive list of 5 problems associated with this fusion theory. Firstly, the putative fusion site on human chromosome 2 suffers the problem of lack of genomic synteny at the molecular level. In other words, there is a lack of corresponding gene content and DNA sequence similarity between humans and chimpanzees at and around the hypothetical site of fusion. If you line up the chimpanzee and human chromosomes, the genes at and around the fusion site do not match. If the chimpanzee chromosomes 2A and 2B are the ancestor origin of human chromosome 2 by means of an end-to-end telomeric fusion, then surely the putative fusion site in human chromosome 2 should have been surrounded by a wide array of genes and DNA sequences with homology to the ends of chimpanzee chromosomes 2A and 2B. But the evidence clearly does not support a case for strong homology, so the human chromosome 2 cannot be the result of an ancestral fusion of two chimpanzee chromosomes with a degree of certainty. Secondly, telomeres confer stability to chromosomes and act as buffers to prevent loss of genetic material. They are typically located at the ends of linear eukaryotic chromosomes and are, at least in part, present to prevent chromosomes from fusion via a capping function. They contain a highly specialized end cap called the sheltering protein complex that protects them from fusion. This suggests that, in principle, telomere-to-telomere fusion should not generally occur. For this reason, telomere-to-telomere fusion is rarely documented in mammals but in states of genomic instability such as cancers, which are not indicative of a healthy state. The prevalent types of fusion are therefore satellite DNA-to-satellite DNA fusions or satellite DNA-to-telomere DNA fusions. Yet in chromosome 2 fusion model, what we have is an end-to-end fusion, that is telomere-to-telomere fusion, which is highly unlikely if not impossible. Thirdly, given the inferred evolutionary timescale of roughly 3 to 6 million years of divergence from a human great ape common ancestor, telomere sequences of TTA GGG repeats at the fusion site are highly degenerate, vague, or ambiguous. One study reported that only 28% of these telomeric repeats matched and the rest had degenerated. But the problem is the timescale of 3 to 6 million years is insufficient for such a high rate of degeneracy. Furthermore, unlike what one would expect of a pristine fusion sequence, the core fusion site of 798 base pairs that is densely populated with the telomeric motifs is not a unique signature, but a genomic sequence motif that is found throughout the human genome without an orthologous counterpart in chimpanzees. It is only about 70% similar to a pristine fusion sequence of similar size. In other words, the fusion site is not as exceptional as made to be, and it is a ubiquitous feature of the genome. It certainly does not support the chromosome 2 fusion model with a high degree of confidence. Fourthly, looking at the comparative cytogenetic image which is drawn to scale, the alleged cryptic centromere site is located at very different locus to what one would expect in a fusion event. This lack of positional correspondence is not restricted to the cryptic centromere site. We also have, to a lesser extent, a discordant positional correspondence of the fusion site. Furthermore, there is an overall size discrepancy between human and chimpanzee chromosomes, which shows about 10% loss of chimpanzee chromosome 2b, that amounts to about 24 million DNA bases. Such a large amount of genetic loss would be catastrophic and fatal to life. Also, the hypothetical cryptic centromere is at best 41,608 bases in length, which is much shorter than standard human centromeres of 250,000 to 5 million bases. And the alpha sequence it contains produced no homologous hits when queried against the chimpanzee genome using two different algorithms. Moreover, at least 6 other regions outside the centromere of chromosome 2 were found to bear these centromeric alpha sequences. This illustrates that the hypothetical cryptic centromere is not as unique as is presented to fit the evolutionary model. Even worse, the hypothetical cryptic centromere contains an actively expressed protein coding gene referred to as ankyrin repeat domain 30B-like, which is abbreviated as ANKRD30BL. This shows that the hypothetical cryptic centromere is actually a regular functional gene rather than a defunct centromere. So the cryptic centromere that is shown to support the chromosome 2 fusion model is not even worthy of being called a centromere. Lastly, it is common sense that the alleged fusion site's location should not be in any life-supporting functional genetic feature, which is highly expressed in hundreds of different cells and tissue types. Functional genes do not arise by mythical fusing of telomeres. But to the irony of Darwinian evolutionists, the strongest evidence against the fusion theory came in 2013, when it was discovered that the supposed fusion site in this case is actually an integral part of the gene called DDX11L2, which was previously thought to be a pseudogene. Instead, this turned out to be a functional non-coding RNA helicase gene expressed in at least 255 cells and tissue types. Moreover, it is expressed in 16 out of 31 major tissue types and therefore is one of many life-supporting genes found in the human genome. The putative fusion site, when read in minor strand orientation, is found right in the first intron or functional start site of this gene, where one of the 3 transcription binding sites is located. So the alleged fusion site is actually a functional promoter element, rather than a blind, unguided, undirected, unsupervised, impersonal and random accident of a historic fusion. It would therefore be a far-fetched and a huge leap of faith to claim that fusions of this kind can lead to functional genes. Now, while it is sufficient to critique the fusion theory with all of this non-exhaustive list of problems in its entirety, Darwinian evolutionists or their atheistic supporters will not leave a stone unturned to religiously downplay or question the credibility of these scientific claims. Their zeal to fit every claim to evolutionary orthodoxy of common descent is often treated as not in need of evidence. If they cannot find scientific counter-evidence of all of this critique in its entirety, they will surely resort to attacking one's personal motivations and pass it off as he is a creationist. Atheists in particular have mastered the art of this rhetoric and appeal to motive when in the state of intellectual ambush. It serves a good deterrent from having to engage evidence. Nevertheless, I will add a caveat that the future may unfold new discoveries that may falsify or further consolidate the fusion theory, and therefore would avoid making any positive claim with a high degree of epistemic weight. I would like to thus conclude with an epistemological point. I am inclined to the view that the current evidence at the site of fusion is not clear enough to make any strong positive claims with a high epistemic value, such as overwhelming, strong, tremendous, conclusive, unequivocal, and so forth. Such expressions are nothing but over-exaggerations of highly speculative and epistemically weak evidence. So considering that there is an unexpectedly small fusion and centromeric size and extreme degeneracy, along with a lack of genomic synteny in the chromosome 2 fusion model, no reasonable person would rule out the narrative that there may not in fact be a fusion site on human chromosome 2. This remains not only conceivable, but also a viable alternative hypothesis. Thank you very much for listening. I'll see you next time.